Hi everyone, thanks for joining us for Lunch Break today. I'm Carrie. Today we're talking with Kindra Crick. Kindra is an artist and molecular biologist from Portland, Oregon, who's fascinated by the human brain. Her installations and multimedia work incorporate drawings, diagrams, maps, and imagery from under the microscope. She was featured in our 2018 exhibit, EmBodied. So today we're catching up with Kindra to learn about her work and what she's been up to recently. If you have any questions that you'd like us to ask Kindra, you can type them into the Q&A box on Zoom, and we'll be answering those at the end. So to start off, can you give us an overview of the mediums and subjects that you use in your work?
Yes, thank you, Carrie. And thank you for inviting me to be here, Julia. This is a wonderful platform, and I will start off. I'm more of a conceptual artist, so I use multiple media. And I'll start with sort of my 2D work that I showed in that EmBodied show with SciArt. And then I'll go through how I move from using drawings and scientific diagrams and maps and images from under the microscope to sort of explore ideas and the unknown wilderness of our mind. So start off. This is a series. This was in the EmBodied show using century-old maps, including the Broadmen Map, which you can see here. This is a diagram taken directly out of a book on consciousness. It even references the chapter that it's in. And I see these as a landscape. And so for the work I was doing at the time, which was on contemplating climate change locally in the Pacific Northwest, I wanted to reimagine this brain, which looked like topography to me. And really using this in the phrenology map and kind of looking at those about how we explore the unknowns in our own mind. And again, the map is not the territory. We sort of use those as a provisional atlas. And so this particular media used all water media because I collected rainwater. And I used wilderness maps from the Pacific Northwest, which you can see on the right, and also another diagram that was researching the past 100 years of glacier melt on this mountain in our region called Mount Hood. And so I took that research in those maps and I cut them up, and you can see here's a process shot where I took the Broadmen's map, which is kind of this classic map of the cerebral cortex. And then I took that wilderness map and I cut it into the different Broadmen areas and then placed them down in a collage and then embellished it with water media, ink, and also material. And you'll notice that a lot of my material the material is a bit of the message. So in this case, I use encaustic, which you have to heat up to almost 200 degrees to make molten. Wow. And you lay over the surface and then it solidifies and then you can carve into it. So kind of further creating this topology over this image of the cerebral cortex that I've now manipulated. So some of these things you can't see kind of just looking at it two-dimensional, but here's Broadmen's wilderness. Again, at the final result with that encaustic layer and mixed media. And the last piece that I'll just show from this series uses another century-old image of the brain, which is the phrenology map, which is now debunked, but was accepted at the time. And again, it would use the surface of a person's skull to sort of predict what their personalities were. And really kind of in hindsight, that's been a debunked theory, but you know, humans are always exploring at kind of, or science is exploring at the edge of our ability to observe. And sometimes things are more obvious in hindsight. In both are looking at, you know, what makes us who we are in terms of our brain and thought, and also in terms of, this is sort of in a nod to looking at climate change, really kind of that understanding will come and we'll kind of understand more in hindsight. And here you can see that I've played with the surface using that in caustic. And I took a torch and heated up the wax, and you can see it actually dripping off of this painting, which of course is in reference to the melting glaciers. So that's this first series that I'll introduce you to. And then next I'm gonna go into a piece that kind of informs a lot of the next work that I'll be showing, but this is a commission. This is the first time I really worked with sculpture. This was commissioned by Cancer Research UK in their project called What's in Your DNA? And it's in the shape of a double helix. And so I wanted to play homage to my grandparents' contribution to our understanding of the structure of DNA and genetics. And so part of it is dedicated to science. You'll see that there's a chalkboard running up one side of it, and that's chalkboard paint.
Oh.
It's on polyester. This is, or sorry, fiberglass resin. This one was 8 feet tall. And then I use pastel and chalk on there. And so one half is dedicated to my grandfather, Francis Crick, who is probably best known for his work on elucidating the structure of DNA, but also you can see references to the central dogma there of molecular biology. And so I took his writings, some of his letters, and I put those in and rubbed them away, and sort of what you can see are images that are taken from chalkboards and letters that he was involved with. Less known is my grandmother, Odile Crick, who is an artist, or was an artist, and she was the first one to draw the image of the double helix,
Wow.
that kind of iconic diagram. And so the other side is this vibrant blue, which is sort of my homage to arts. And it has these structures on them, which are viral or look like they're seated, and they kind of spiral up this double helix. And this is, in my mind, a metaphor for ideas. So not only do humans pass on their genetics, but we also pass on our ideas, and we do so through the arts in writing and communication. And those are infectious. So this was auctioned off at Christie's in London, and an anonymous buyer purchased it and donated it to the MRC. So you can see it here in the lobby. And this is actually the location where my grandfather did his seminal work on genetics. So that you'll see sort of the imagery show up later in my work that came from that one commission. So also at the MRC was Aaron Klug's work. So this is one of the images that was taken in the 1970s that I use as a reference for sort of these viral images in my work. I like the way they look, and I started using them as a metaphor for the infectious nature of ideas. So this is an image taken in the 1970s. And so you'll see that kind of show up in different parts of my work, but they're really kind of this exploration of ideas and our mind. So in the Chalkboard Memory series, again, I start off using the surface of a chalkboard. So I paint panels with chalkboard paint, and then I work the surface. And this was to explore memory and that fragility of memory, sort of what ends up on the surface. And so I would create marks in chalk and actually pastel, because it has pigment in it. And I would erase and rewrite that surface. And I actually pull on my collaboration, which I'll talk about later, into memory, but this idea of memory gets reconsolidated, and it is changeable, just like the surface of a chalkboard. And then I also sort of slightly like it as this metaphor again for the scientific method, that through iteration, we can get to kind of what is most useful to us. And I'll just call out the quote that I decided to keep on the surface here, which I got from a scientific article, by I love that the scientist used a metaphor to describe memory, which was that memory is the neural echoes of experience.
That's beautiful.
And I also use things that are laying around my studio. So I just happen to have a slide deck that my grandfather used. And I remember this lecture he gave. He was a neuroscientist when I knew him. So he would give lectures and he would use optical illusions to kind of call out, you know, how our visual cortex or how our visual system worked. And so this Necker cube I really like, and I incorporated into this piece about perception. I love that it's a static image, and I don't know which face the viewer is going to see as being forward, and it will change, kind of calling to attention to this idea that the viewer actually has a role in terms of how they perceive any given work, and it can change and it can flip, just like this Necker cube. And this last piece I'll do in this series just speaks to kind of my obsession with sleep. I love this idea that sleep and memory are intertwined, that, we, record things, somehow they're mapped in the hippocampus and they're kind of broadcast out to the cerebral cortex, and there's this reconsolidation happening. And so you can kind of see this in my work here. And again, the chalkboard, this idea that forgetting is a component of memory, and that sleep is important for that. All right, I'm gonna quickly switch gears here. I find collaborations to be stimulating. I was selected to be part of the Jordan Schnitzer printmaking residency at Sitka. So this picture is of the Oregon coast where the Sitka Center for Arts and Ecology is. And I did a printmaking residency there. And here they specifically chose non-printmakers to be paired up with Julia DeMauro, who's a master printmaker. And so for that project, because it was printmaking, I really wanted to dive into this series that I had been kind of in my notebook, this Infectious Ideas series. You can see this woman, she has these viruses and bacteria that are kind of coming out of her head, potentially to go on and spread. And I wanted to use the printing press because in order for ideas to spread and mutate, they need to be copied. And the printing press itself was a huge part of this historically, that allowed plays and poetry and manifestos to be copied and spread throughout the world. So this is called Infectious Ideas. It's an intaglio print, hand-dyed, or hand-colored ink. And then I'll just quickly show a few others from this same residency. I'll call attention to the middle one. This is called Tangled Memories. Again, I use images of neurons from under the microscope, and here I use this soap ground in order to put it into this silhouette, and it's this kind of nice organic tangle that you see there. And now I'll get to sort of more recent work and collaborations, how I got from two-dimensional work to full room installations. This really started with a Northwest Noggin collaboration with Dr. John Harkness. You can see here, he's a researcher who works on memory and cocaine addiction. And we're at the confocal microscope looking at thin slices of mouse brains, which are stained to reveal a structure, which you can see here, which are perineuronal nets. And they are not part of the neuron, but here you can actually see them. They're kind of in the shape of neurons because they're this extracellular matrix, this netting that's around the neuron that is thought to be important with neuroplasticity. So our ability to form memories, keep memories, and potentially change memories. And when we were giving a talk together, there was no place for two-dimensional work. And I really loved a description that had been made of it called describing these nets as corsets. So I decided to use fabric. So this is split wire cables, magnet wire, LEDs, fabric, and then this kind of netted shimmering fabric to kind of call attention to this little detail of these nets that potentially hold our memories together. And so we showed it, a piece of it, at the Phillips Collection in DC, and then I brought it back here and created a full room installation because I wanted people to be able to actually walk through a cross section of the brain and be able to see these images of these neurons, these biological machines that might make up our memory. And when I did this installation, there was sound, which you can hear here, done by Katie Greisner. And there's also a projection on the wall showing some of the images from under the microscope. And so this was shown at the Lippmann Gallery here in Portland. But I wanted to make something that you could only see under the microscope, larger than life, and something that you could have a full immersive experience in. And so this is another shot of that. And then elsewhere in the gallery, there's this 10-foot sculpture, which is dedicated to the father of neuroscience, which is Ramon Cajal. You can actually see his drawing on the left here. And he cataloged neurons, the whole nervous system, including neurons in the brain. And so this is called In Memory of Cajal. But I wanted people to be able to interact and kind of play with these dendritic arms lit up by these LEDs. All right, in this last installation here, this actually was shown up until January at Washington State University, where the research on perineuronal nets was done. And here I stripped away the sound and the projections and the LEDs, and I just created a crown of these larger than life neurons connected together with this fabric, so that the viewer could see them above their head. And in addition to that, also sort of thinking about kind of the modern day Cajal, I created portraits of individual human neurons, which have been cataloged by the Allen Institute, and painted in India ink and sort of metallic ink on hand-dyed kozo paper to sort of immerse and surround the viewer in that space. But we have 86 billion neurons that make up our memory and who we are. And I wanted to kind of call them out as individuals and portraits and have them sort of dancing around the gallery. And so here's just an installation shot for scale. But that was my most recent project that came down in January.
Yeah, thank you so much for an overview of the many different forms that your work has taken. And it's beautiful to know the backstory of your grandfather with the science and your grandmother with the arts and how that's melded into your work. This is obviously such a strange time to be creating and researching, but would you tell us if there are any ways we can keep up with your work, see what you're up to, and follow along with what you're doing?
Sure. So one of the projects that I've been involved with actually for the past 5 years was that Recycled Rain project. And Sarah Scholl, who's the curator, actually decided to do it again this year. So for all of April, and actually a little bit of March, I was collecting rainwater for this project. And because of this kind of stay-at-home time, I wanted to go back. And so you can see that it's online until May 31st. But during this time, I wanted to sort of reflect on sleep, because my sleep had been interrupted. And so I went back to a notebook where I had taken notes and done some drawings from a lecture given by Harvard sleep scientist Robert Stickgold. So that was in 2017, I think. And he had gone over REM sleep and dreaming. And so I looked at the image that I had drawn in that notebook and some of the notes, including one that said, "In sleep, we hold tight to what's important." And I thought, oh, that's a really great sentiment. So I took the sketch that I had worked in there. It was a woman who was upside down, had these neurons coming where her hair should be. And there was a moon, I think, to indicate that it was night. And so I used pen and ink, and in this case, gouache and watercolor, using that rainwater I collected, and created this imagery. And in REM sleep, this is where Stickgold said insightful and sort of connections were made. So I wanted to kind of put the image of the fantastical. And then also I like to put into my work, I don't think I showed this too many times, but I've done it before, but in the night sky that she's laying over, there are these constellations here. And so I will put in neurotransmitters so that if you look deeply, you can see that the constellations in the sky are actually
Oh, wow.
which rises during dream sleep. So there are little things that I like to put into my work that reference specifically and directly, different neuroscience aspects of things that I incorporate. So that is this piece. And then another place that I like to post my work on Instagram, so you can actually see me, a fast forward of me kind of creating this piece on Instagram. I do eventually update my website, so you can find a beautiful video done of the illuminated wilderness by Ian Lassaro up there, kind of a professionally done one. And then I do outreach with Northwest Noggin, which is an arts integrated neuroscience outreach group. And so if you go there, you can see some of the projects I'll be working on, including sleep. And of course, now I'm very much into circadian rhythms. So hopefully some of that will show up in my work soon.
Great. Thanks so much. We'll now be taking some questions from the audience. There's still time if you have a question to send that in. And Julia's going to hop in.
Okay, Kindra, thank you so much for sharing your work. It's wonderful to see what you've been up to recently. So one of the questions we have in is, how do you feel your understanding of neural chemistry and memory affects or interacts with your experience of memory in your life and work?
Well, in studying memory, I think, especially in relation to sleep, I'm very aware of if I want to do something. So for example, supposedly if you want to remember something, or it's been shown that if you want to remember something, you should sleep afterwards. So for example, in getting ready for this presentation, I looked at everything I wanted to remember, and I thought I should do that right before I go to bed. So there's some things that kind of are practical. I think the other thing which is kind of interesting about knowing the neuroscience of memory is also understanding how inaccurate your memory can be. So for example, if someone were to ask me a question about why I did something 10 years ago, I sort of have this understanding that part of me is slightly making up what I think now, about something 10 years ago. And since the context has changed, my 10-year ago self might be surprised about what my current answer would be. So.
That's great. Yeah, I feel you there. Another question in, do you think your art has affected the everyday way you see the world? Do you see patterns like you depict in your art more often around you now?
Well, I think that when I use patterns, especially those sort of seed-like viral patterns, I both collect them. So I think I even have something in my studio, which is like an allium, which is back there. And then people sort of gift them to me because they know of my interest. So I definitely am interested in sort of the fractal-like patterns or tree branching of neurons. And so I do look for what I would consider sort of biomimicry. So again, looking for those patterns. Oh, I enjoy organic chemistry. I enjoy constellations in the sky. So I do sort of collect those pairings. So it's something that I see under the microscope and then what that might look like in the real world. So yeah, I do definitely think that I seek them out.
Great. One question I had was that I was noticing kind of as we progressed through your work that the color blue pops up more and more. And indeed, your shirt is some shade of blue. Can you talk a little bit about that? I'm sure you're aware of it.
Yes. So this is actually, I almost was referencing that answer in that previous thing, as if you asked me 10 years ago that question, I might answer differently. But now, because it's been called to my attention, I've now created an answer for that.
That works.
There are a few that I enjoy. Like, I'm like, oh yeah, sure. One of them is that I had this wonderful conversation when I was younger with Christoph Koch. Actually, I was part of a conversation, and my grandfather, and they had a whole 3-day long conversation about what was the blueness of blue. And as like a 15-year-old, I thought, oh, that's pretty simple. And realized by the end of it, I had no idea what the blueness of blue was. And we could describe everything from our understanding of wavelengths, et cetera. And so that was something that was always stuck in my mind. And then the other thing is that there's actually been a shift over time. So I was interested in the opsin. So one of the components of optogenetics responds to blue light. So you can take something from a single cell organism and you can put it into a neuron, and then you infect the neuron, and you can actually get neurons to fire using blue light. So I thought, oh, that's wonderful. I'll use that in this installation sort of to reference optogenetics. And then most recently, with my understanding of the circadian rhythm, one of the opsins, so melanopsin in our eyeball, a receptor, is actually attuned to blue light. And that actually sort of communicates with your suprachiasmatic nucleus and kind of tells you, oh, you should wake up, or this is, you should be responding to the day. So it's kind of changed over time, but I have always sought out, I guess, blue as different pigments in my work. So that's a really long answer, but it's evolved over time.
Yeah, that's wonderful. It's so funny how I feel like every artist has, one or two or three things that they end up just coming back to again and again in different ways, and you don't always know why. But of course, like your hindsight piece, you look back on certain things that have happened or certain things you've read about, and you're like, oh, of course I'm, attracted to color. Of course I keep bringing it back in because it's a theme throughout your work. It's a thread.
Right.
But yeah, that's so funny. So well, that is all the time that we have for questions. So thank you so much, Kindra, and I'm gonna let Carrie wrap us up here.
Yeah, thank you. This has been really fascinating. And thanks for everyone who joined us. Thanks for your questions. So our next lunch break will be Wednesday the 20th with Justin Levesque. And this Friday, instead of lunch break, is Laser Boston, which is a virtual event that will have 3 speakers across the arts and sciences. If you missed any part of this talk, it will be posted on YouTube afterwards, so you can check that out there. And you can join our mailing list at sciartinitiative.org to stay up to date on what we're doing next. Thank you so much again, Kindra, and we'll hope to see you next time.
All right.